All right, so now we're going to be doing macromolecules, and this one is very content heavy, not a lot of processes really. We're going to be doing definitions, so this one is another PowerPoint slides. I don't have much to draw, but you're just going to have to sit down and kind of look through these and get comfortable with them. They will come back in almost every class after this: Anatomy 1, Anatomy 2, Pathophysiology, and Microbiology. So you will see them again. Nutrition, if you haven't already taken it. So it's a good little intro. So lipids is our first macromolecule we're going to talk about. There's four main ones we discuss, but lipids is the one category that we will not consider a polymer. So the other three, so your carbohydrates, your proteins, and nucleic acids, we consider those polymers because they're made up of monomers. But lipids are not polymers because there is no monomer. We don't have that building block. So with that, most lipids are hydrophobic. So think back when you see the word hydrophobic, does that mean it's water insoluble or water soluble? If we were in person, I would ask you to answer, but we're not. So the answer is water insoluble. If it's hydrophobic, it means it's afraid of water. It has a phobia of water. So with that, it's not going to dissolve in water since it's going to be afraid and avoid it. So again, water insoluble or hydrophobic. So the function of lipids include to store energy. They make up cellular membranes. They make up that phospholipid bilayer. You can see in that top right picture, that is your whole cell membrane, the whole bilayer, and that's made up of phospholipids, which is under the lipid category. And then they also make up a lot of hormones. And you'll see some examples of hormones in your notes. So what are our different types of lipids? First, we have triglycerides. This is the main one you'll see. Triglycerides are the number one lipid kind of seen throughout the body. Triglycerides, the tri meaning three, they have those three tails, a glycerol and three tails. It's a long-term energy storage, and you'll hear that in adipose tissue, especially in lab. You'll look at adipose tissue, and that's where we store energy. And it also builds up very quickly. We have to work out to get it off. And then it provides structural support, cushion, and insulation. So again, that structure there, you see the glycerol on the left and the three fatty acid tails hanging off, making it the triglyceride appearance. All right, phospholipids. This is what I kind of talked about with the phospholipid bilayer. So a phospholipid has a polar head and a nonpolar tail, meaning one region of it is actually hydrophilic, it lacks water, and one region is hydrophobic, meaning it hates water. So when you have any kind of molecule that has both sides to it, we call it an amphipathic molecule. So amphipathic means it has both. Your cell membrane forms the chemical barriers of the cell. So your phospholipids make up that cell membrane, which protect the cell. It has a hydrophilic head. So the head is what's facing the water, and then the hydrophobic tails, which are just two fatty acids hanging off that polar head. And those are hydrophobic, meaning they don't like water. So if you see in that picture, you have the polar heads facing in towards the cell and out of the cell. And that's what likes water. So it's in contact with water. In the middle there, the nonpolar tails are hydrophobic. So they are facing away from water because they're scared of the water. So they face each other. All right, steroids. So steroids, big thing here is they make up cholesterol, steroid hormones, and then some bile salts. I wouldn't get too caught up in the structure, but it's still important to kind of just make sure if you see it, you would know it. And it has 4 carbon rings, but I would really know that they make steroid hormones. And then eicosanoids, this is a modified 20 carbon fatty acid. And then mainly it has signaling molecules, makes up signaling molecules, and it has some functions in the inflammatory response, blood clotting, and the nervous system communication. Really important are that inflammatory response and blood clotting. All right, moving on to carbohydrates. So carbohydrates are a polymer. Polymer meaning it's made up of monomers. And monomers are those building blocks. So our monomer for carbohydrates, which is really important, are monosaccharides. Think about mono meaning kind of one. So monosaccharide, the most common one we'll talk about is glucose, and that's how we supply energy for cells. So monomers to monosaccharide, but we don't just get all forms of glucose already down. When we eat things, we have to break it down. So normally when you eat something, it starts off as a polysaccharide, which is multiple monosaccharides put together, and those are glycogen, starch, and cellulose. Glycogen is the storage form of glucose in animals. So anyway, once you break down a polysaccharide a little bit, you are left with a disaccharide. A di meaning two means two, so monosaccharide plus monosaccharide. And your most common ones are sucrose, lactose, and maltose, and those are just combinations of certain things like maltose. It's just 2 glucose molecules together. So once we break up a disaccharide even further, we are ending with a glucose, which is aka a monosaccharide. So a bunch of monosaccharides together, and we have a polymer. So nucleic acids are responsible for your DNA and RNA. So with that, the monomer for it, since it is a polymer, it's a nucleotide. So this picture on the right here, that whole thing is one nucleotide. So the nucleotide is composed of that phosphate group, that little pink circle on the left, the pentose sugar, or the sugar, which is that purple in the middle, and then the nitrogenous base, that little blue square to the right. So with that, the sugar depends on what you're looking at. If you're looking at DNA, it's deoxyribose nucleic acid. That means that the sugar is deoxyribose sugar. With RNA, ribose nucleic acid, you're looking at ribose sugar. So the nitrogenous bases, there's several different ones. So here's a little concept map to kind of show you the breakdown of them. So with nitrogenous bases, we have two main categories. We have pyrimidines and purines. Pyrimidines break down into your cytosine, your uracil, and your thymine, and your purines break down into your adenine and guanine. The way I remember it, I don't know if you've heard of the Purina dog food or animal food. I think about Purina, the dog food is meant to feed animals. Animals are agriculture. So I think of ag, so purine and ag. So purines are the adenine and the guanine. And then for pyrimidines, think of like in elementary school when you cut pyramids out of paper. So cut cytosine, uracil, and thymine, you cut pyramids, so they're pyrimidines. You'll notice under uracil and thymine that they're in specific structures only. So adenine, guanine, and cytosine, you'll find in DNA and RNA. But uracil is the version of thymine in RNA, and thymine's the version of uracil in DNA. So they kind of switch out when you're switching from DNA and RNA. So types of nucleic acids, the first one is deoxyribonucleic acid, which is your DNA. So DNA is double-stranded. Think of the D for DNA as double-stranded. And that is your genetic information. So it determines what your proteins are going to look like and how the protein should be synthesized. And proteins are everything in your body in terms of function. So DNA stores that code for that. You can find it in your chromosomes in the nucleus, and you can find it in your mitochondria. Mitochondria has its own set of DNA, and it can divide and all that kind of stuff, which is really cool, but we won't get into that right now. And then the monomer is your deoxyribose sugar, your phosphate, and nitrogenous base to make up that nucleotide monomer. So ribonucleic acid is RNA. It's single-stranded. Just think deoxyribose nucleic acid is double, so RNA is opposite, single-stranded. It also helps with genetic information. It's found in the nucleus and cytoplasm. It's in that cytoplasm because that's where it actually goes to the ribosomes to actually synthesize proteins. And we're not going to get into that right now because we have a whole unit on it. So we'll come back to how DNA is made and how RNA gets out into the cytoplasm and to those ribosomes to synthesize the proteins. And the monomer, which is a ribonucleotide, is made up of ribose sugar, phosphate, and then nitrogenous base to equal that nucleotide. And then one you'll see throughout the whole entire semester is ATP, which is adenosine triphosphate. And this is how we get energy for cells. So it is a ribose sugar with 3 phosphate molecules attached and an adenine. So again, we have that building block, that nucleotide. ATP is extremely important, and we'll talk about it more as we see it throughout the semester. All right, so proteins, this is our last big section, and it's a polymer as well. So again, if it's a polymer, it has a building block. That building block for this, the monomer, is amino acid. An amino acid would be one of those little red chains in the bottom picture. So the GLY, that one alone would be an amino acid. When we start connecting amino acids together in chains, that's how we start building a protein. So amino acids are connected one by one via peptide bonds. So when you see peptide, think P, protein. That's how we connect amino acids to make a protein, with those peptide bonds. So between each of those little red circles there in the bottom right picture, every single one of those is connected to the next one by a peptide bond. So with that, there's several different structures we can have, and it's kind of based off the name. So a dipeptide, di meaning 2, it means 2 amino acids together. Oligopeptide, this is kind of our small group, and that is 3 to 20 amino acids. Polypeptide is 21 to 199, and then protein is anything 200 or above. And yes, I would say to memorize those. But no, if it's anything above 200 or above, always go protein. If they say there's 500 amino acids together, you know it's a protein. So when we start building proteins, we start adding amino acids onto each other. But we can't just have one long chain of amino acids. It doesn't work. We start to fold them, and folding them into certain structures is how we get their function. Shape equals function. So when we first start putting amino acids together, we're going to start with the primary structure. The primary structure is just a linear sequence. It's like taking them and just putting them together in a straight line. So let me draw here. So put amino acid there, connect it with the peptide bond to the next one, keep going in this linear sequence, and that is a protein. Not a protein, but that's how we're starting building the protein, because there's not quite enough yet. So that's a linear sequence held together by peptide bonds. Now we move on. Once that starts getting longer, we need to start folding it some more. So we hit our secondary structure. And once we hit secondary structure, that's when we actually hit 3D realm. So we hit that 3D shape. And the conformation is what we call the change of shape. And so in order to actually start folding it, I think it's a really fun word. You start to have chaperones. So chaperones are what help assist the folding. So they help with that shape change, that conformation. So chaperones are the ones that help and monitor the folding process. So with the secondary structures, you're going to see two different things. You're going to start seeing alpha helixes and beta sheets. So alpha helixes taking it and kind of intertwining them together. So let's draw. So it'd be like taking one and start combining them together. Beta sheet, you start folding it. So take one strand and start folding it into sheets. And that's your beta sheet. Both are condensing it, so making it smaller and taking up less space, but also starting to have a shape to it in order to have a function. All right, once we are moving on from that secondary structure, we're going to move on to tertiary, third structure. And that's your final 3D shape. This is the actual one protein is put together. We have a full protein chain. There's two types of tertiary structures, globular proteins and fibrous proteins. Globular proteins, they're exactly how they sound with the glob. Think of a glob of something. It's just a whole jumble, a jumble of proteins together, but it's not a random jumble. It all has a shape specific to its function, but you just need to know what they look like and what their definition is. Fibrous, I think of like a fiber being a thread of something that's more linear. So this is still a glob, but it's not just a big circle. It's in linear shape. So it's a fibrous or a fiber. And that's your fibrous protein. So that's the end of that one protein structure. So primary, secondary, and tertiary structure. However, we can combine tertiary structures together in order to make bigger proteins. And we see that in the quaternary structure. So the quaternary structure, this is when two or more protein strands combined. And all you're doing is taking some globular proteins and combining them, fibrous proteins, combining them, or some of both. So we would have one globular protein, and let's get a different one, different color. That did not change color very well. It's not changing color on my screen. But if you look at the picture, you can see the teal, the orange, the red, the green, they all combine, and those are all globular proteins mixed together. All right, so I'm gonna clear these drawings, and that's the end of that shape for the protein. So here's a practice problem. If we have two amino acids together, so what classification would this amino acid be? Would it be a dipeptide, an oligopeptide, a polypeptide, or would it be a full protein? It would be a dipeptide since we only have two amino acids. All right, so now next practice question, we have 30 amino acids. So 30 amino acids, where would that fall in? Would it be a dipeptide, an oligopeptide, a polypeptide, or a protein? And that would be a polypeptide. All right, so denaturation. I'm not quite sure why the disease there. Yeah, denaturization. I say denaturation is in the picture. I'm not sure if it's either way, but denaturation, that's just when you change the shape of a protein. Remember, function of a protein is based off its shape. So if you change the shape of that protein or denature it, like start unwinding it, unfolding it, it disrupts that normal function. So it's rendering it useless in a way. So denaturation occurs in several things. One would be an increase in temperature. That's why if you start to get a really severe fever, like over 106, and start denaturing proteins, you start to can lead to serious consequences, including death. Increases in pH or decreases in pH. So if our blood became too acidic or too basic, it could start denaturing proteins. So we never want to have a big increase or decrease in pH. One thing I will say though, because some people get confused here when it says increase and decrease in pH, that they think the temperature is the same. So they think an increase or decrease in temperature would denature it. Decreasing the temperature does not denature the protein. It once it gets cold, it's not really changing the shape or denaturing it. It slows down chemical reactions, but it's not really changing the shape of the protein. So with this list, remember these three, but remember a decrease in temperature for this class, we're not going to talk about it in terms of denaturing a protein. So please do not think that on the test if it comes up. It's these we have written down here. All right, so the protein functions, this is straight memorization. Protein functions, there's several of them. First one is it can serve as a catalyst, and a catalyst is just anything that speeds up a reaction or changes the activation energy required to start a reaction. And we'll talk about that in its own unit, so don't stress too much about it right now. Proteins act in defense, so that antibody down here, control, this little Y-shaped thing you'll learn about in anatomy too is an antibody, and it is a protein based design that helps with the immune response and binds to antigens, things like that. Next is transport, your membrane proteins that you talked about. So the sodium potassium pumps, the leak channels, gated channels, all of those things are based off protein. So that is an actual protein channel. It helps with structural support. So protein, for example, in your collagen, things like that, make up skin, nails, muscles, protein and muscles, all help with structural support in the body. They cause movement. So again, muscles, you have proteins responsible for movement. We'll spend a whole test on muscles alone. So you'll see that whole cross bridge cycle come back with all the troponin and tropomyosin. So again, don't stress about that yet, but just know it causes movement. It helps perform regulation. You have proteins with performing regulation in the body. It helps with fluid control, things like that, because proteins help pull. And then they provide storage. So proteins help with storage. And that's really it for the protein functions. We're not going to cover metabolism in this video.